We're going to talk about the different types of selection that there are. And we're going to be looking at several graphs. So we're going to see graphs like this. Graphs have two axes. The horizontal axis is called the x-axis, and the vertical axis is called the y-axis. In this graph, the horizontal axis represents the body size of organisms in your population. So you have organisms with a body size of 125 grams and all the way to 25 grams. And the y-axis represents the number of organisms. So here at the peak, that means you have the highest number of organisms are at this body size of 75. While if you look over here at 25, there are very few organisms with 25 grams of body size. In this other extreme, 125, you have very few organisms at that side. And most of the organisms in your population are within this range of about 60 to 90, let's say. Let's look at how selection can change the distribution of traits in your population. If selection is favoring individuals with either large body weight or low body weight, we will have what is called disruptive selection. So here the individuals with the mean body weight are selected against, so they're least favored. They are less likely to survive and reproduce. And most of the organisms that survive and reproduce are either in the low body weight or at the high body weight. Now you will have a population that looks now like two curves, where most of the organisms either have low body weight or high body weight. And this is what is called disruptive, because you start with one population that is very homogeneous, and now you have what appears almost to be like two populations. And if selection keeps going this way, you could end up with two populations that could eventually even become two separate species. Another type of selection is directional selection. So in this case, you're favoring either the individuals with a high degree of the trait or a low degree of the trait. Looking at this example of body size, if you favor individuals with the largest body size, then over time, generation after generation, the mean of your population would shift, and now most organisms in your population will have a higher body weight average. You will have more organisms in this higher range of body weight. It could be the opposite direction and still be directional selection. Let's say you're favoring individuals with the low body weight, then you will see the shift will happen in this direction. You will have most of the organisms will be in the low body weight. And finally, the third type of selection is stabilizing selection. In this case, the favored organisms are the ones that are closest to the mean. So what would happen here is that the mean will stay the same, but now you have fewer organisms that are on the lower end of the range or at the higher end of the range. So you have sort of the compressed variation of your population. Let's look at some examples of these types of selection. We see stabilizing selection in the weight of human babies. Most babies are born within a very narrow range of body weight, and those that are in that range have the highest chances of survival. Babies that are born with lower weight have fewer chances of surviving, and babies that are born too heavy also have lower chances of surviving. There is selection favoring babies that are born in the very narrow range of body weight. Directional selection can take many forms. Let's say you're selecting flies to avoid light. They're more likely to go into darkness. After multiple generations, you will see that most of the flies in your population prefer darkness over light. If instead here of having body size, we have preference for darkness, you would see that over time, generation after generation, your population has shifted to preferring darkness over light. And this could be the same case with many traits. For example, humans selected corn to have more kernels. So over time, the number of kernels in an average corn cob has gone from what it used to be, and now you have a higher number of kernels. An example of disruptive selection is what we see with the finches in the Galapagos. These finches have two sides of beaks. They have large beaks that they can manipulate large seeds, or they have small beaks that allow them to manipulate small seeds. The original population that colonized that island probably had a distribution like the one here in pink, where the majority of the organisms had a medium beak size, few had larger beaks, and few had smaller beaks. The ones in the middle were selected against because they weren't very good at manipulating large seeds or manipulating small seeds, while the ones with large beaks were very good at manipulating the large seeds, and the ones in the low end were very good at manipulating the small seeds. If this continues, you might end up with two separate populations of finches, one with large beaks and the others with small beaks, that could even end up being separate species. And in some cases, selection actually increases genetic variation. Up to this point, we've been looking at examples in which selection mostly narrows down the variation or moves the genetic variation in one direction or the other. But there are cases in which selection increases genetic variation. Ways in which this happen are frequency-dependent selection. The negative frequency-dependent selection is when you're too common, you're less likely to reproduce. And positive frequency-dependent selection is when you're too common, then you're more likely to reproduce. Oscillating selection is when the direction of selection changes, and this is for external reasons. So you might favor individuals with large body size sometimes, and then other times you would favor individuals with smaller body size because something in the environment is changing that is favoring different traits at different times. And the last one is the heterozygous advantage. This is when there is an advantage of being heterozygous for a trait, but being homozygous for either one is at a disadvantage. Of these water bugs, the percent of water bugs that are eaten by predators, and how frequent their color is in the population, and the different colors of bugs that there are. And what we see is it's not a matter of which color is more likely to get eaten. You see that all the colors are likely to get eaten, but it depends on which color is more common. So if the dark brown color is the most common at that time, that will be the one most likely to get eaten by predators. But if the light brown color is the most common one, then that will be the one most likely to get eaten by predators in that time. In this case, the most common phenotype is the one that is most likely to get eaten. So that means is the least likely to survive and reproduce, so that the rare phenotypes can now become more common and you have this negative frequency dependent selection. Positive frequency dependent selection is the opposite. Here is when if you're common, you're more likely to survive and reproduce. If you're rare, then you're less likely to survive and reproduce. So this fish, the one that stands out is the one that is most likely to get eaten by the predator, so it's the one less likely to reproduce and pass on those rare genes. Oscillating selection is when the direction of selection changes depending on the environment. An example are the finches that we talked before that have smaller beaks that can eat smaller seeds and that have large beaks that can eat larger seeds. So when in the Galapagos Island you have El Niño and La Niña events that can bring years of drought and years of excess of rain. When you have a lot of rain, there is a high production of small seeds in the island. Finches with the small beaks are favored because there will be a lot of small seeds for them to eat and they will produce more offspring so that the offspring will inherit that small beak trait. But when you have years of drought, only the large trees are surviving. The large seeds are the ones produced, and the finches with the large beaks are the ones surviving and producing offspring. Offspring produced in those years are mostly descendants of the large beak finches, so they have also large beaks. And this is changing from year to year depending on whether it's a rainy or a drought season. So we're shifting the population back and forth between more of the smaller beaks or more of the larger beaks. And since this is not dependent on the frequency of that phenotype, it's not about which beaks started as being more common, it's rather about the environment, what conditions the environment has that favor one trait over the other, and this is changing from year to year. This is called oscillating selection. So it's similar to frequency-dependent selection in that it changes back and forth, but it's not driven by how frequent the phenotype is. It's rather driven by external conditions in the environment. Finally, the heterozygous advantage. This is when heterozygous are favored, but being a homozygous dominant or homozygous recessive is disfavored. An example of this is sickle cell anemia. Individuals that are homozygous dominant, they have normal red blood cells, but they're also very susceptible to malaria. And in areas where there is a lot of malaria cases, these individuals are likely to get infected with malaria, and this could mean death. On the other hand, individuals that are homozygous recessive for sickle cell anemia will have these oddly shaped blood cells, and they will have anemia, which means they cannot carry enough oxygen to their tissues, so they will be seriously affected. The heterozygous are somewhere in the middle. Their red blood cells are not completely affected. They don't quite have anemia, but they have the advantage of being resistant to malaria. But this advantage is only for the heterozygous. Those that are homozygous recessive are severely ill because of anemia, and those that are homozygous dominant don't get any protection for malaria. So this means that natural selection is favoring the heterozygous, but when you're favoring the heterozygous, you're increasing the frequency of the recessive allele, and when you increase the frequency of the recessive allele, you're also increasing the percentage of homozygous recessive.